it isn't just the sequencing data that is growing, right? We have data coming out from the sequences, the genomes, then the expression studies, the proteomic studies, metabolomics for that matter, microbiome, the methylation, and the exposome. So, there's a whole field of omics with an explosion of information. And then there's the play of this information among each other.
I
So,
want to
this.
hear this now.
Could we please take care of that?
Yes, ma'am.
Yeah, there's also systems, and so now we are coming back to a systems biological approach, a holistic look at what these biological systems are doing. We're getting a greater understanding of these. And it's not just biomedical sciences for that matter. I was application scientist at Agilent Technologies, and I spoke to researchers from a lot of fields of biological research. It wasn't just the human systems or diseases. I spoke to people, agricultural scientists, people working on transgenic rice. I spoke to people that were working on single cell bacteria. I spoke to people working in soil microbiology, and they are all using these high throughput systems to generate data at a really fast rate, because there are questions that they want to answer, and they want to understand these processes as to how they network among each other. Now, all of these present a lot of opportunities. This is high throughput data. There's huge amounts of it. It's growing explosively. We need clean data, and we are getting data of multiple types, as I've mentioned already, from proteomics, from microbiome, etc. What are the methods that are required? You need to store these in efficient systems. You need efficient algorithms to actually mine this data. Since these data sets are huge, you need your methods to be scalable. You want to keep your signal to noise ratio low, so you want these methods to be accurate. And because there are of multiple types, you need to know how your genes are interacting with your proteins, what kind of metabolites are being produced, how is the bacteria in your gut microbiome interacting with them or producing them, what kind of pathways are involved. So you need these algorithms, these methods to be integrative. We can think of all of these as challenges, or we can all think of them as opportunities to innovate. And it is in this landscape, that the bioinformatics as a field is born and is continuing to develop. It is truly an interdisciplinary science. It is an integration between life sciences and computational biology. It is a field that develops and applies computational technologies to study biomedical questions, and its roles are twofold. As a technology, it is a powerful tool to manage, query, and analyze big data in life sciences. And as a methodology, it is top-down, holistic, data-driven, genome-wide, system-wide approach, and it generates new hypotheses. It finds new patterns and discovers new functional elements. So if truth be told, we're looking at a fully integrated system which complements traditional biological experimental methods. It is a very seamless way of combining the computational technologies with the experimental biological systems. And it very nicely overlays with medicinal and clinical informatics systems, biology, and synthetic biology. So now we know what bioinformatics is. We're moving on now to how the bioinformatics helps. If you look at this slide, it talks about biology in bioinformatics. So we are going to be discussing the biological questions informatics allows us to study. The framework remains genotype to phenotype, and it is within the bounds of the central dogma, which says DNA is transcribed to RNA, RNA is translated to protein. What we see here is sequence alignment, looking at genetic sequences of two genes, maybe from two different species. Are they homologs of each other? Are they similar? And if we know the function of one gene from one species, can a homolog of that gene in a different species serve the same function? Does it inform us of the functional value of that particular gene? These sequence alignments in the at the DNA level allow us to search for genes. They allow us to compare different species and allow us to study how evolution happened. They tell us about the DNA methylation of these particular genes. When we are looking at the RNA, we are looking at the differential expression in different conditions, disease and good health, in different species, in different populations. We're looking at co-expressions. We're looking at which are the ones that are coding, which are the ones which are not coding. And when you look at the protein level, you can identify a lot of unknown proteins as well from your mass spec. And proteins are never linear, right? They are folded into different structures, and these networks allow us to study the protein structure as well and predict the protein structure. And again, inform us if you know one protein from one species, we can maybe guess at the structure of that similar protein in a different species. None of these things work completely, absolutely in isolation. They form molecular networks. And we get more information about protein networks, transcription networks, how they're regulated, and the metabolic and signaling networks, the signaling pathways, how our hormones, interact with each other. We're able to simulate what a single cell will, how it will be able to function. And this allows us to compare different populations. Ashkenazis use the Alaskans. They're always used to, the efficient model systems for a lot of diseases. It allows us to do that. When we move on to the informatics part of bioinformatics, again, the framework here is from data to discovery. There are a lot of computational methods that are required to manage, search, and analyze data. We've already seen just how huge our databases are. So laptops these days, they have a good memory. Some of them are 120 GB, some of them are greater than that. However, one single NGS experiment, right, terabytes of data, it cannot be on one laptop. We need different, proper servers, systems, databases to store that kind of data. To actually mine information from these data, we need computational tools. And these computational tools are allowing us to create networks between different genes, between different proteins, between the metabolites. And they also allow us to model, and we can create predictive models. We can simulate systems. And this is how bioinformatic tools are enabling discovery process. I hope I've been able to kind of help you understand why bioinformatics is so important. I will end with saying that bioinformatics is central to life science research at a molecular level. Thank you.